Hello again, everyone. Welcome to the second episode of this series of educational webinars. Last week, we talked about brain connectivity in source space. Today's topic is M and EEG beamformers and virtual sensors. I am Harold Bornfleth, and I'm responsible for the BESA product portfolio. I am again joined by my colleague Jae-Yan Cho, who is actually the mastermind behind the implementation of the time domain beamformer at BESA, and who will assist with questions and answers today. So, what's on the schedule today? First of all, we are introducing the topics. Then we will look at a bit of theory. Then we will see the beamformer in action and try out different beamformer flavors on some data, using the BESA research program as a vehicle. Please, throughout the demonstration and the webinar, put your questions in the questions area or in the chat boxes, and we will try and answer as many of them as possible at the end of this session. So why should we do beamforming? The beamforming mechanism is a unique way of solving the inverse problem in MEG and EEG brain imaging. For each brain voxel, a new inverse operator is computed. This is like in dipole localization. These voxels can then be scanned over the brain to produce a brain image, and this is like in distributed volume imaging methods. And the inverse operator can be applied as a spatial filter on raw data. For example, like we do it in the dipolar source montages, but with the additional benefit of inherently contrasting against background brain activity. So this makes the beamformer voxel act like a virtual sensor. Beamforming is a spatial filtering approach to source localization and was introduced to the field of MEG-EEG by Van Veen and others in 1997. Differences to other methods. The main thing is that not just the lead field matrix, but also the data covariance matrix is taken into account for computing the beamformer spatial filters, or the spatial weights. We are always looking at one particular location in the brain for which we estimate an activity. This is our virtual sensor, and we can then scan this location over the source space. And finally, activity is contrasted against either a reference interval or a reference condition. For example, I want to contrast the activity in a time interval after a stimulus with the activity in the baseline. Then I need the following ingredients to compute the beamformer image value. I need the lead field at a source location. I need the data covariance matrix for a signal interval. These two give me the beamformer spatial filter at a source location. And applying the spatial filter again on the data covariance interval, this gives me the power for the signal interval at a given source location. And now I can do the same for a reference interval, which is, for example, the baseline interval. This also gives me the power for the baseline interval for the same source location. Now I divide the power over the signal interval by the power over the baseline interval. And I have the final beamformer image value at my source location. And then, scanning over the complete source space, I can compute the beamformer image over the brain. In mathematical terms, this looks quite complicated. So I only have this one slide with mathematical terms on it, I promise. So let's start with the data covariance matrices. These are computed from the samples in the baseline and in the signal interval and all the trials. Then we also have the channels to consider. And we also have the lead fields. For a location R, we compute the lead field in the three directions of the current flow For EEG, this will be X, Y, and Z. For MEG, we would only use 2 tangential orientation vectors. From these, we then compute the beamformer spatial weights for the location R. We use the lead field and the inverse of the covariance matrix. This gives us spatial weights W for the signal covariance and also spatial weights WB for the baseline covariance. Now we can apply the spatial weights. For the beamformer image, we use the contrast of the output power of a signal interval to the output power of the baseline interval, and then we repeat this for all the source locations. And this is how we get to a beamformer image like this. If, however, we apply them on the actual measured data, we can also compute a timeline for this location R. And this is the virtual sensor scenario. So for any measured data, we can compute the source waveform. So let's look at this in a little bit more detail. We have a spatial filter at a given source location. We multiply it with the measured data. This gives us the source waveform estimate for this source location, i.e. we have a virtual sensor at this location in the brain, and in the image on the right, down here, you can see where the sensor is in the brain, and on the left you can see at what latency it is active for the 3 different orientations X, Y, and Z of the current flow. Right, that's great. Let's stop and have a coffee. But unfortunately, there's a few caveats. So one thing is that the beamformers have a problem with a highly correlated activity in different brain regions. But okay, problems are there to be solved. So one solution is a so-called multiple source beamformer. It simply includes more than one location in the formula for computing the spatial weight. BESA research uses a bilateral beamformer. So that takes homologous sources in the opposite hemisphere into account. And the result looks like this. For an auditory evoked potential, as an example, the left side shows a single beamformer scan over the brain, and the right side shows a multiple source beamformer scan, in this case, a bilateral beamformer scan. Obviously, in this scenario, the bilateral beamformer has a huge advantage in separating the activities of the 2 hemispheres, as you can see here in these 2 example images. Then another thing is the regularization. This is something that haunts all inverse methods. In order to compute the inverse of the covariance matrix, some regularization needs to be applied, because we usually have less individual components in our data than channels. We can try to invert without regularization, but that will lead to an amplification of noise. So what happens when we increase our regularization parameter? This will decrease the spatial resolution of the beamformer image. On the other hand, it increases the output power. And if we decrease our regularization parameter, the opposite will happen. You can see here an example where the left shows a case of 1% regularization and the right is the same data with 10% regularization. So you can see how regularization gradually smears the signal over more brain areas, but it may also enhance some of the signal. In the BESA software, you can switch between multiple source beamformer and single source beamformer. What you can also do is to switch from the scenario of having the 3 orientations of the current in your beamformer to having just one orientation, hopefully the correct one. This is called the scalar beamformer because we look at just one direction rather than 3 directions. And the formalism for this was established by Robinson and Verba and Sekihara and others. Yeah, and there are also some other potential improvements for the beamformer result. There was an idea in the community to not completely separate the baseline interval and the signal interval, but also look at the common interval that spans the whole activity. You can see this common interval in this little picture here in blue. So below here, you see the standard way again of putting together the beamformer results. So we have a lead field and covariance matrix for signal interval at the top. We have a lead field and covariance for the baseline interval at the bottom. And they're contrasted against each other. But what if we relax this condition and we give the operator more leeway on which covariance matrix should be used to compute the spatial filter? So instead of having the fixed scenario of the signal and the baseline covariance matrix, we could just say, well, maybe there's another one that helps us to find out a better spatial weights filter. So let's have a covariance matrix of choice for our estimate of the spatial weights, maybe both for the signal and the baseline scenario. For example, we could decide that we were only interested in the signal interval for the spatial filter. So in this case, we could simply discard the baseline interval for covariance matrix for calculating the spatial filter. We only calculate one spatial filter from the lead field and the signal interval covariance matrix. And that same spatial filter is then applied on the signal covariance data to calculate the power of the signal, and on the baseline covariance data to calculate the power of the baseline. And in another approach, we can use the common interval that spans the whole time range of activity. So again, we only compute one spatial filter for both signal power and baseline power value. However, this signal covariance matrix and the baseline covariance matrix, they're still relevant for both these approaches, since we multiply the spatial filter with them to calculate the actual power values. And finally, another variation to the theme is to use noise normalized weights for computing the spatial filter. And this goes back to ideas of Sekihara and Cheney and their co-authors. If we use this approach, we can even compute a beamformer image for each time point in the data, as was shown here in this little video. So it will not just be one image computed from the whole signal interval, but we can actually differentiate between activities at different time points. Good. So these are variations on the beamformer theme. And last topic for the introductory talk before we look at some real data application is the topic of virtual sensors again and their relation to source montages. I mentioned earlier that the virtual sensors can reconstruct the time course of activity of a brain source. This is the same idea that we have in Bezier source montages. And this slide here shows you how in a traditional, so to speak, source montage, you have to ensure that each brain region which has activity is modeled by a source. The example here shows the following. So we have activity in both temporal lobes. If I only place one source in one temporal lobe, it will also model some signal from the contralateral temporal lobe. So here in this signal reconstruction, you see an overlap of signal actually from this temporal lobe and signal that comes from somewhere else. If we, however, place two sources separated, one in each temporal lobe, then we can see that the signals from both lobes can be separated. And now we can see that one signal is actually earlier than the other one. In this case, the red one peaks earlier than the blue one. And I can also put additional sources in the brain at other locations to check if these brain regions have activity. In this example here, they don't. So in the upcoming version of Bezier Research 7.1, which is due to be released shortly, we will offer atlas-based source montages. So for several brain atlases, we have a montage available where dipoles or regional sources or beamformer virtual sensors are placed into the center of each atlas region. You can see here in this example the visual stimulus signal that is part of this experiment here mainly activates these green areas which locate in the occipital part of the brain. And this source montage can then be applied to the raw data. So instead of looking at waveforms at the sensor level, like here on the left-hand side, you can see the activity directly at the level of the brain sources, like here on the right-hand side. Yeah, as I said, by distributing the sources of the whole brain, I can be fairly sure that my sources show activity really from their locations and do not model activity from other remote areas of the brain. And now if I use beamformer virtual sources instead of this, or just maybe a few beamformer virtual sources, instead of this large number of sources distributed over the brain, I should in theory have an advantage, because the beamformer spatial filter is already constructed in a way that excludes contributions from remote parts of the brain. Okay, and this is maybe enough theory for the moment. So I think now it's time to try the beamformer out in real life. So this is the BESA research program, which I'm going to use as an example to show you the effect of these different beamformer options. And we're going to start with an auditory experiment because it's quite instructive to show a few things. Here we have just 32 scalp channels, and we have an experiment with different intensities of tones. So there was low intensity tones and high intensity tones, and some were intermediate between low and high. And they're distinguished by different trigger codes, 1, 2, 3, 4, 5. So throughout the data, these stimuli were applied, and we can now define the paradigm for this and use it to compute beamformer images. So I'm going to click this ERP push button. The actual definition of these paradigms is part of another tutorial, so I will not go further into this. We have already defined these conditions for different stimuli between 60 decibels and 100 decibels intensity. We have epochs that are throughout the range between 300 and 1000 milliseconds around the stimulus. We always take slightly longer baseline and post-stimulus epochs than maybe other people would, because that gives us an option to also do post-hoc filtering without disturbing signal of interest. We have performed an artifact scan. So this scan here shows you the trial information. So each trial is depicted by one column in this, and how they behave over the different channels. So the channels are the rows, and you can see, for example, there's 2 channels that have very high signal in many of the trials, and I can use this slider to sort out trials that I don't want. These are the ones that are in red, and the ones that I keep, they are the ones that are not red. So I can scroll through this and see that most of them are actually kept, and only the worst ones have been sorted to the right and will be excluded. So that's good. I can also check the gradient criterion and see that there's not much problem there. And then I can go to the average tab, and here I can define conditions to average or conditions to run the beamformer on. And I can select the low condition here, which I can see has 124 accepted trials. And I can use the beamformer button now to invoke a beamformer scan on this. If I do this, then I get to another dialog box that allows me to tell the program about the intervals that I'm going to use for beamforming. And it's easier to use the butterfly plot. So when I click this, I get this overview of this condition over all channels, and I can see the intervals also depicted in different color shadings. So let's look at the signal interval. I want to work around the N100 and N150, so I'm going to use the 80 to 150 interval, so we can see how the red interval slightly adapts when I change it here. I can then go to the baseline and adjust it accordingly. So it would make sense to have a similar length of baseline compared to signal interval for the beamformer to work best. So I'm going to enhance the baseline interval a little bit. So to make it from minus 120 to minus 50, we can see that that still seems to include sort of real baseline type data, not too much noise there. And the common interval should then span both these intervals. So I'm also going to adjust that. The common one is the blue interval now. And now I'm set up to start the beamforming. So I'm going to click this OK button here and press OK here. And now the source analysis module of Beza Research starts. This is working with an average brain because I haven't co-registered a brain to this data set. And this is already the result of the beamformer. So we can see that there's 9 maxima that were found by the beamformer scan. And I can scale the display up. So I can see that the first maximum is actually very close to the auditory areas where I would have hoped to find it. And I can scan through the maxima using this M button in the toolbar. So there's another one slightly back here, another one. So I can then go to different maxima. I can see I also get a contralateral maximum in near the other area of the beamformer. So this, yeah, was an okay result for this type of scan. And I can now see if I can improve the results by adjusting some parameters. So let me go minimize this window and I get, again, the foreheads view like this. And I can use this arrow here in the center area to open a pop-up and go to settings. And this should allow me to change settings for the beamformer. So I've got the regularization pane here, and beneath that I can see the options for the beamforming. And by default, yeah, Beza uses this multiple source beamformer. So that was why I was able to actually separate these auditory areas. So let's just, for comparison, try the single source beamformer. And I'm going to apply the settings and press go, and this will then start the beamforming. And if I leave the dialog, I can go back and find the central maximum, and I can see that this is the typical result of the single source beamformer for a highly correlated signal like the auditory signal. So it will actually get the signal into the center of the brain rather than finding the homologous auditory regions. Good. Other settings we can influence are, for example, the use of these different covariance matrices. So let me go back to the settings, and instead of using the standard beamformer, I'm going to use just the common weights. So remember, the formula then means that the spatial filter will be created from the common interval rather than individually from the signal and the baseline interval for the two filters that I want to contrast. And let me try this. So again, I'm going to apply and press go. And I should also go back to the multiple source beamformer. So apply, press go. And now I can see that if I go to the maxima again, I actually have an improved solution in comparison to the first one. So I have again 7 maxima. We go make it large. But I think that they are closer to the auditory areas, and also the second maximum now already has the roughly homologous location in the contralateral hemisphere. So That's what I would have expected from the auditory potentials anyway. So I can see that this change of putting together the spatial filter has actually improved my result. Okay, and the next thing I would like to do is create a so-called virtual sensor from this beamformer result. So I have the option of going to any point in the brain really, but it kind of makes sense to use the maxima. So I'm now on the first maximum, and beside this maxima toggle button, I have another button here that allows me to insert a source at the current crosshair location. So if I click this one, then I can see that now I've started a solution in the program, and I have inserted this virtual sensor at the beamform maximum, and it gives me directly information about the time course of the signal. Then I switch to the next maximum, and I can do the same thing and also insert the maximum here. Yeah, so now I have my two virtual sensors in my two controllator areas that already reconstruct the time course of the signal. And I can also apply this to the raw data, or really also the average data of my auditory experiment to see how the signals are transformed from the sensor level to the brain level. So to do this, I can save it as a source montage. So I'm going to save this under the, S1 is my file. So call it beamformer test, and then close my source analysis module. And I can see that this source montage is now applied on the raw data, and I can use the options button to check whether these sources will show all traces, so all three, or whether the program tries to orient them in the best way to explain the signal on the screen. But I think safest bet is to show all the traces. So, and I can now browse the data and see how the signal gets transformed into this source space using these two virtual sensors. Well, one thing that could be interesting now is to actually see them applied to the average data case. So let me open the average data file. So this is the S1 average. And for this one, I can now also apply the same source montage that I've just created using my virtual sensors. And I can see that these averages are actually performed over different intensities from 60 decibels over 70 and so on. And we can see that in the baseline, there's not much activity, and then getting gradually stronger over the, so once the intensity rises. So it seems like it separates the activities quite nicely, but how about comparing that to a traditional source montage? So it would be good to see, is there any advantage, or is it the same thing as the Bezier source montages that we've already had for 20 years? So let's just go and quickly model another example. So I'm gonna just go over this data set low. So this is my low intensity average here, and perform a source analysis on this one. I'm just gonna do a slightly reduced interval from -100 to 250 milliseconds. And I'm going to have a filter set between 0.5 and 40 Hertz. Right, and now I can perform source analysis just by placing sources, converting them to regional dipoles. So regional sources are more robust, and then in terms of an auditory data set should help me to find a fitting solution more easily. And also I can constrain the location of the second auditory source by making it symmetric to the first one, so that'll make it even more robust. So now I can see that the second one will be following the first one. So let me just fit these over the main interval, all fit, start fit, and have a look at the anatomy. So this is actually a co-registered data set. So I can see this one was fitted very closely to the Heschel gyrus, and the other one actually looks also quite good in shape. So this simple fit worked pretty well, and I can now define a source montage with just these two sources and save my source montage as S1 regional sources fit. Okay, and this is already applied now to the raw data, so to the, not the raw data, but to the average data. So I can see this is the result of the regional sources fit, and I can now toggle between this one and the beamformer source montage. So, and I think you can actually see that the beamformer picks up less noise than the regional source montage, even though it was a very good fit of the regional sources. So the regional sources, especially when we get to higher intensities, we can see that there's a lot more going on. And in particular, when analyzing this experiment in more detail in another tutorial, we figured out that there is a third source that comes into play at high intensities, and this disturbs or overlaps with the primary auditory sources. So, and here we have a problem with the traditional source montage because it doesn't model all the active brain regions, whereas the beamformer source montage contrasts against other brain regions, so it isn't so strongly affected by this. So here we do have an advantage of this virtual source scenario using the beamformers. Good, and now I would like to come to one more aspect of the beamformer, which we also briefly touched upon in the introduction, looking at the time capacities of beamformer images. And I would like to change the example for this. So I'm going to work with an MEG example, which I've also used last week in the tutorial. It's the visual motion example. So let me have a look at an MEG example. First of all, let's just look at the MEG channels. Here we have 121 MEG channels, gradiometer channels, and we have two trigger codes, 1 and 2, which code for motion stimulus. So that was a point cloud moving towards the spectator, and a static stimulus, which was simply a static point cloud. And this example also has a paradigm associated with it, which is, let me just check that we have a co-registration first. So Control L brings up my co-registration dialog. So I have a co-registration with the individual brain here. Yeah, and then I go to the ERP module again, and we see there's two conditions, motion and static. So no surprises there. And we're going to go to the artifact scan to see if there's any artifacts that need to be rejected. So this was a very clean data set. Actually, we don't have many problems. Let me just exclude maybe this one and then go on to the average. So we still have 84 and 88% of our trials available. So I'll just mark this condition motion here and then go to the beamformer tab, and I can do the same approach as I did with my auditory data set. Look at the butterfly plot. Okay, so the butterfly plot shows me there's the main signal starts at about 100 milliseconds. Okay, this is already adjusted because I've done it once before. So here we have the 100 to 190. This should cover all my main activity, at least in the first part of brain activation, and I've got a matching baseline length, and I've got a common interval that spans both of them. So pressing OK will then start the beamformer calculation and start the source module and show me the result. So here we are. This was the beamformer calculation. It's already there, and I can scale it up. This was the maxima. So I can toggle through. I can also toggle back using the control key. I wanted to stay with the first one and then scale up a bit. So the primary activation is in the primary visual areas, and then we get to more lateralized areas. And now I can again look at the settings that I can influence. So I can also use the shortcut image settings here from the menu. And currently I've got my common weights enabled, got the vector beamformer, and now I can actually look at the noise normalized weights and apply and press go. And this will then enable me to do a scan over the brain that also allows me to see how the brain image changes over time. So, so for now, set a cursor here. Then I can move this cursor around and see how the brain image changes over time. I hope that comes across. So scale up a bit more. What happens in this experiment from initial stimulus, and then there's another peak in the global field power that we can see here. So going back to the initial activation, we can see that here in the primary visual areas, we have the highest activation. And now we move out and then we get to a second maximum. And we can see very nicely now that the activity moves to lateralized areas here. So you can see that at this latency, which is 165 milliseconds after the stimulus, we now have a lateralized activity, not so much anymore in the primary cortex, but in the motion detection areas of the brain. Okay, and again, now we can exploit this by defining virtual sensors and then applying them to raw data and do all kinds of things like connectivity analysis. Maybe if you joined last week's webinar, I was talking about connectivity also with this example. We could now actually insert, this is of course not what I wanted. I want to go to the maximum of this. Switch this off. Delete the source and insert this source instead, and then move to the next maximum. And I was actually going to find another lateralized one, insert a source on the contralateral side, and then we can go back here and go to the primary activation and find the maximum of this one, and also insert a source at this position. So now we have a very nice approach of accounting for our differential activation at different times, even in the same dataset using a beamformer virtual sensor. And I could now apply this as a source montage and then do connectivity analysis on these virtual sensors instead of a source montage if I wanted to. So let me just make this an example. Test beamformer. And we can see how these sources now, and for the MEG we only have two orientations, so each source now has two time courses and how it's applied on the raw data here. So yeah, quite a useful thing, I would think, and this is actually most of what I was going to talk about. There's one more thing I would like to show you a glimpse of what we've been working on recently, and that will bring me to a similar topic. So talking about source montages, virtual sources, virtual sensors, and also brain atlases. So let me close this program and just want to show you, this is the beta version of our latest release that we are currently finalizing. And if I use the same data set, then yeah, I need to run this beamformer again to get actually to the beamformer choices. So let me just run this. This should be pretty quick. So now we can also see the intervals in the top left here. So we can see this where my baseline signal and common interval was. Of course, this is what we saw before. And now I can do one thing. I can open a solution here and need to go to the montage folder. And for example, for the brain atlases that we have implemented in BESA, we now have solution files that cover the centroids of the different atlas regions, and we've got them available as beamformer sources, virtual sensors as regional sources, and radial single dipoles. So loading this then shows me this beamformer result. I just need to make sure before I apply it that I change to the right type of beamformer. It would only make sense if I use noise normalized weights here. So let me just actually switch these sources off for a second, then run the settings with noise normalized weights, run the beamformer again. Okay, and now switch them on again. Okay, and now if I go through the data, then I can see how my brain beamformer sources individually change activation over the source space. So I can see this in my average data here, but I can also apply this again as a source montage. So just going here, press source montage as, and with this beamformer calculation, I will use the spatial filters from the beamformer calculation to apply them to the raw data. So I'll call it ALLBF noise norm. And that is now applied to my raw data here. So now we can see all these AAL atlas sources are applied to their raw MEG data. So again, this could be some way of going from your sensor level to source level without any prior knowledge of where your sources have to be, just using a standardized atlas to apply this to your sources. Good, and I think now you've listened to me long enough. This brings me to the end of the talk. Just now I want to thank especially Jae-Yan Cho, who was the mastermind between the implementation of all these beamformer options, and yeah, who's also on the call with me. And there was also Mateusz Rosiniak, who worked on the brain atlases and the atlas source montages, and Michael Shirk, Patrick Burke, Nicole Ille, and my late colleague Dieter Weckesser. They all were instrumental in getting the source montage functionality to work. Thanks very, very much for joining me in this webinar, and hopefully talk again. My colleague Mateusz Rosiniak is providing a webinar talk this Thursday about artifact correction and joint fMRI EEG recording, which I think is also highly relevant for many research applications. And then next week is the final webinar in this series about Bayesian source imaging. Thanks very much again, and hopefully talk soon. Bye.